So if you look at a human tumor under a microscope, specifically, let's say, non-small cell lung cancer or ovarian cancer, you are just looking at a lump of rogue cancer cells.
Right, not at all. You are looking at a massive swarming army of your own immune cells.
Exactly. But for decades, scientists have been haunted by this one single question. How did they get there? And, how did they know what to do? Did those immune cells get recruited locally and trained right there on the site? Or did they arrive with pre-programmed orders from somewhere else?
That is the central mystery of cancer immunology right now. We've debated it for years, largely because, well, we just haven't had the technology to track their movements in living human patients.
Which is exactly what we're digging into today. Welcome to the deep dive. Our mission today is to exhaustively unpack how scientists are finally tracking the hidden lives of innate immune cells in human cancer. We are looking at a really fascinating preview piece from the journal Cancer Cell called Attack of the Clones.
It's an incredible paper. The team led by Lou and colleagues really cracked open a huge barrier in tracking these cells.
Yeah, and to appreciate that breakthrough, we kind of have to understand the barrier first, right? Because we've been tracking adaptive immune cells like T cells pretty easily for a while now. They have what basically amounts to natural barcodes.
Yeah, exactly. T cells and B cells undergo something called somatic recombination.
Right.
So during their early development, they essentially cut and paste their own DNA to create unique antigen receptors. That shuffled genetic sequence acts as an endogenous barcode. So if you find two T cells with the exact same genetic sequence in that receptor, definitively they're clones born from the same parent.
But the innate immune cells, like the monocytes, the macrophages, the dendritic cells that act as your frontline infantry, they don't shuffle their DNA like that. They don't build those highly unique receptors, which means, well, no natural barcodes.
None. And in animal models, like mice, we can engineer our way around that. We use genetic fate mapping. Things like Cre-recombinase, which acts as a kind of molecular pair of scissors,
Yeah, so you can artificially insert fluorescent tags into the DNA of specific progenitor cells in a mouse's bone marrow, right? And then literally watch where those glowing cells end up.
Exactly. We just track the glow.
But obviously, we can't genetically engineer human cancer patients just to satisfy our scientific curiosity.
Right. Ethics boards tend to frown on that.
Just a bit.
Yeah.
And relying on standard somatic mutations, the random, everyday DNA typos that accumulate as human cells divide, those are just too sparse. They're too rare to reliably trace a cell's lineage over a lifetime.
Yeah, so researchers needed a completely different tracking device. If the nuclear DNA is a dead end for tracking these innate cells, they had to look elsewhere.
And the team in this paper turned to a really groundbreaking technology called MT-SCATAC-SEC. It's a mouthful.
It is a massive acronym.
Yeah, but let's break it down, starting with the first part. The MT stands for mitochondrial.
Which is such a brilliant pivot, because instead of trying to find incredibly rare mutations in the massive genome of the cell nucleus, they just looked at the mitochondria. Mitochondrial DNA mutates at a significantly higher rate than nuclear DNA.
Right, it happens fast enough to act as an alternative naturally occurring barcode. So when a cell divides, its daughter cells inherit that specific mitochondrial mutation signature.
Exactly. It creates a highly traceable family tree. But that's only half of it. The second part of that acronym, the ATAC-SEC, that's not about tracking where the cell came from. It's about figuring out what the cell is actually doing right now, in real time.
Because ATAC-SEC measures chromatin accessibility, which is basically you have to picture how DNA is physically stored in the cell. It's not just floating around loosely, right?
No, it's tightly spooled around proteins.
Yeah, so if a cell needs to use a specific gene, it has to physically unspool that section of the DNA so the cellular machinery can actually read it. ATAC-SEC uses an enzyme that only tags these physically unspooled open areas of chromatin.
Precisely. It gives us the literal blueprint. It shows us which biological instruction manuals are currently lying open on the desk, ready to be read.
And by combining these two techniques in single cells, you are mapping the cell's ancestry via its mitochondria and reading its current behavior via its chromatin, all at the exact same time.
Which is an incredible innovation, but, and this is a big but, it comes with a massive technical hurdle.
Right, because if we're looking at human solid tissue, getting cells out means breaking up the tissue. Doesn't mitochondria just sort of spill everywhere from bursting cells?
You hit the nail on the head. That's ambient mitochondrial contamination, or horizontal mitochondrial transfer, if you want the technical term. When you physically dissociate a mass like a lung tumor, you inevitably rupture some cells.
Like popping a water balloon.
Exactly. And their unique mitochondrial barcodes just spill out into the fluid. That ambient genetic material can then get scooped up by perfectly intact cells.
Oh, wow. So that would completely ruin the data. You'd think two cells were siblings when they actually just, bumped into the same piece of biological debris.
Right. Your entire family tree would be built on lies. Which is why tracking these lineages in solid tumors has been so agonizingly difficult.
So they needed a fix for that, an empirical computational framework.
Yes, and they called it Mitotrek. Mitotrek essentially acts as this ruthless bouncer for the data. It rigorously filters the sequencing and strips out anything that even hints at ambient contamination. It demands absolute, high-confidence clonal assignments.
And it's so strict that they ended up only capturing about 23% of the cells they analyzed. They threw out over 3/4 of the data, just be sure.
It sounds wild, but it ensures the integrity of those family trees. And you have to look at the scale here. Even after tossing all that data, they still had an astounding 218,715 cells.
Across 10 patients, right?
Right.
Looking at non-small cell, lung cancer, and ovarian cancer.
Yes, across 3 distinct compartments: the matched tumor, the non-involved healthy tissue, and the peripheral blood.
Okay, so let's do a quick 5-minute checkpoint for you listening at home to lock this in. First, human innate immune cells lack natural tracking barcodes. Second, this myeloid tech uses mitochondrial DNA mutations as an alternative barcode. And third, the Mitotrek algorithm cleans up that spilled mitochondria data to give us high-confidence cell lineages across over 218,000 cells.
That's the perfect summary.
So a question for you listening to mull over. If you could drop a microscopic GPS tracker on any single cell in your body from birth to death, which organ cells would you want to follow?
Oh, that's a good one.
Right. Okay, so let's get back to the behavior. Let's use an analogy to visualize the data here. When T cells, the adaptive immune system, identify an enemy in a tumor, they act like local special ops, right?
Yes, they lock in and clone themselves rapidly right there on the battlefield. It's an antigen-driven local expansion.
But the innate reality is totally different.
Completely different. The Mitotrek data shows that myeloid clones, the innate immune system, are small, and they are phenotypically heterogeneous. They aren't a local militia. They are a systemic circulating infantry.
Unpack that word for me, heterogeneous. What does that look like in the clones?
It means a single family tree is a mixed bag. You might have dendritic cells, macrophages, monocytes, natural killer cells, all coming from the exact same parent.
Oh, wow.
Yeah, the data point that blew me away was that within clones of 5 or more myeloid cells, the mean fraction of actual myeloid cells was only 58%.
Wait, only 58%? So nearly half the cells in that family tree weren't even specialized myeloid cells.
Exactly, which means the parent cell back in the bone marrow had to be incredibly versatile. But the distribution is even wilder. 27% of these myeloid-dominant clones spanned all 3 tissue compartments.
Meaning you'd find one sibling in the blood, one in the healthy tissue, and one fighting in the tumor.
All at the same time, compared to just 8% of CD8-positive T-cell clones doing that.
So what you're saying is the tumor isn't forcing these myeloid cells to multiply on site. The immune system is just constantly parachuting in fresh troops from the bone marrow.
Precisely. The myeloid compartment in the tumor is sustained by continuous replenishment from the periphery, not local proliferation.
That makes so much sense biologically because the tumor microenvironment is incredibly toxic, right? It secretes immunosuppressive chemicals. If your innate cells only replicated locally, the tumor would just suppress that entire local bag.
Exactly, would shut them down immediately. By deploying from the blood, it's an evolutionary safeguard.
Okay, time for our 10-minute checkpoint. Let's recap. 1. Myeloid cells are constantly replenished from the blood, not multiplied in the tumor. 2. Myeloid clones are incredibly diverse, spreading across blood, healthy tissue, and tumors. And that leads to a question for you. How does knowing your immune system is fighting cancer as a whole-body, systemic war, rather than just a local skirmish change how you view treatments?
It completely reframes it. You're treating the whole supply line, not just the front.
Right. Okay, so in this systemic war, the paper focuses heavily on a very specific VIP cell, type 3 dendritic cells, or DC3s.
Yes, DC3s are vital.
Why do we care about them so much?
Because they are the communication link. They are responsible for priming tissue-resident memory CD8-positive T cells.
Ah, so those are the exact T cells you want activated to actually fight and kill a tumor.
Exactly. You need DC3s to present the targets and give the kill order. But for years, there's been this massive debate in immunology about the lineage of DC3s. Where do they actually come from?
Right, the two suspects. Do they come from classical dendritic cells, the CDC2s, or do they come from monocytes?
Right, and without this mitochondrial lineage tracing, it was mostly just educated guesswork in humans.
But the Mitotrek data finally solved it.
It did. By looking at how often these cells co-occur in the clones, the researchers found DC3s are clonally linked much more closely to monocytes. They share a barcode with monocytes way more often than with CDC2s.
But they also had the epigenetic proof to back that up, right? The ATAC-seq data.
Yeah, the chromatin accessibility data is the nail in the coffin. DC3s have open chromatin at regulatory elements that are usually associated with monocytes and macrophages, not classical DCs.
So it's basically like looking at the finished house of a DC3, checking the foundation, and realizing the concrete was clearly poured by a monocyte contractor.
That is exactly what it looks like. The structural blueprints match. And what's great is that this aligns perfectly with recent mouse fate mapping studies.
Oh, right, the animal models.
Yeah, they showed that DC3s come from a monocyte dendritic cell progenitor, an MDP. Now, the exact immediate precursor in humans still needs some functional follow-up, but the origin is clear.
The human body creates these highly specialized intermediate cells like DC3s, instead of just using standard off-the-shelf monocytes everywhere.
Because specialized jobs require specialized tools.
Okay, our 15-minute checkpoint. Number 1, DC3s are vital for activating anti-tumor T cells. Number 2, they are born from the monocyte lineage, proven by both shared barcodes and epigenetic blueprints. So a question to you listening. Why do you think the human body bothers to create such highly specialized intermediates instead of just letting basic monocytes do everything?
It's a great question about efficiency versus precision.
Right. And speaking of precision, this brings us to the ultimate question of this whole deep dive, the most pressing mystery. Who gives the orders?
Yes, the big one.
Does the local tumor environment dictate what a blank slate monocyte becomes, or does that monocyte arrive with pre-written instructions?
To figure this out, they looked at the shared clone experiment. They found sibling cells, exact same lineage, where one went to the healthy, non-involved tissue and the other went into the tumor.
The perfect control group.
Exactly. And the finding was stark. In healthy tissue, a clone stays a basic monocyte. In the tumor, the exact same clone family differentiates into dendritic cells or macrophages.
So the local tumor environment absolutely provides the go signal to transform.
It pulls the trigger, yes. But, and here's the aha moment, among the cells that do differentiate, if a clone becomes mostly DC3s in the tumor, its rare differentiating siblings in the healthy tissue also become mostly DC3s.
Wait, really?
Yes. The fate was already decided. It's a pre-established bias.
Because if the healthy tissue and the tumor both result in the same specialized cell, the environment isn't writing the instruction.
Exactly. The tumor just says execute, but the orders were already in the envelope.
And they proved this by looking at CD14 positive monocytes that were still just circulating in the blood, right?
Yes. By examining the chromatin of those blood monocytes, they found specific transcription factor motifs that were already wide open.
Transcription factors being the proteins that basically control the master switches for cell identity.
Right. And this is where it gets incredibly specific.
Let's give our listeners some memory hooks for these, some mnemonics to remember these complex transcription factors.
Good idea.
So for DC-biased monocytes, the ones destined to become dendritic cells, they have open motifs for IRF, STAT2, and NFE. So think ISN. I see now. Dendritic cells are the eyes of the immune system, right? They see the threats and show them to the T cells. I see now. ISN.
I love that. IRF, STAT2, NFB.
Perfect. And then for the macrophage-biased monocytes, they have open motifs for AP1, CMAF, and NRF2. So think ACN, all-consuming ninjas, because macrophages are the cells that eat and consume cellular debris.
All-consuming ninjas, AP1, CMAF, NRF2. That actually makes it so much easier to visualize.
Right. So just to summarize that subtopic, monocytes carry pre-written epigenetic orders, biases in the blood. The tumor just provides the trigger to execute those orders.
Exactly. The ISN or ACN blueprints are already open before they even reach the battlefield.
Which leads to my final question for you listening. If our blood is full of pre-programmed immune cells carrying their future blueprints, could a simple blood test one day tell us exactly how our immune system will react to a specific tumor before we even treat it?
That is the dream. Because if we can read those orders in the blood, could the next generation of immunomodulatory therapies actually rewrite those orders?
Wow, rewrite them before the cells ever reach the site of inflammation.
Yeah. But it leaves us with this massive, mind-expanding question from the source's conclusion. We know these pre-programmed biases exist in the blood, but why?
Right. Are these biases the result of distant signals from the tumor, echoing all the way back through the blood to the bone marrow, like the tumor is remote controlling the supply lines?
Or is this just the natural, inherent diversity of human monocytes, and the tumor just acts as a filter, recruiting whatever happens to be passing by?
It's a complete paradigm shift.
Yeah.
We now have the first clonally resolved human atlas of innate immune cells. We know they are systemically deployed, and their fates are written in their epigenetics before they ever touch a tumor.
It's truly a new frontier for cancer immunology.
It really is. Well, thank you so much for joining us on this deep dive into the hidden lives of your immune cells. Keep questioning, keep exploring the science, and we'll catch you next time.